We will continue our discussion about this TrkB transport to the cell surface with the help of Calnexin. In the previous video we have understand that Calnexin helps in the transport of TrkB from endoplasmic reticulum to the cell surface. In this video we will discuss what makes the Calnexin to transport TrkB from the endoplasmic reticulum to the cell surface. So from the literatures, the researchers have found out that the phosphorylation of Calnexin will cause partial release of these antitrypsin proteins. So from that they made a hypothesis that phosphorylation of Calnexin might cause or help in the transport of cell surface. Phosphorylation of Calnexin might help the transport of this TrkB from endoplasmic reticulum to the cell surface. So what they did is they create different construct, one that has Calnexin as a wild type and another one that has this Calnexin where it has been mutated at serine at 543 and serine at 544 with alanine, and this correspond to the CK2 phosphorylation serine sites. And there is another Calnexin construct where this serine at 563 has been mutated with alanine, and this correspond to MAPK kinases phosphorylation site. So and they use or inject this construct in a Calnexin deficient cells where they observe the effect of this EGF on this TrkB and other signaling pathways. And what they have found out is that on the Western blot it might not be clear, but what they have found out is that in the case of this CK2 phosphorylations Calnexin construct and the wild type construct, it is able to rescue this cells are able to rescue this TrkB transport from endoplasmic reticulum to the cell surface, and these bands can be seen here. And however, when this construct, that is Calnexin MAPK kinase phosphorylations is been mutated, in that case the intensity or this TrkB, mature TrkB, that is 130 kilodaltons, mature TrkB of size 130 kilodaltons are not been able to rescued, or the intensity is not as equal to this wild type, or with this wild type, the intensity is not been equal to this wild type cells with this EGF inhibitions. And when they and these things that they got it from the quantification of this Western blot that has been shown in this panel B, where we can see that when the Calnexins in the Calnexin depleted cells, when this EGF is used, then there is no TrkB transport, or TrkB transport is minimum. However, when this EGF is used along with this construct, that is Calnexin wild type, they can see the increase in this TrkB transport, and which is almost equivalent to the TrkB transport to the cell surface when this CK2 phosphorylation construct is used. This means that this phosphorylation sites are not important for this TrkB transport. However, when this Calnexins mutated at this 563, that is MAP2 phosphorylation Calnexin sites, in that case they can see the reduction in this TrkB intensity. So from this they are able to find out that it's the MAPK kinases phosphorylations in the Calnexin or the serine phosphorylations at the 563 sites is important for the transport of TrkB to the cell surface. In order to validate these results further, they did the immunostaining, where they can see that In the case of this Calnexin positive cells or wild type cells without any EGF introductions, they can see the cluster formations clearly. However, when the EGF is introduced, they do not see any cluster formations. Oh, let me correct myself. In the case of these wild type cells, where the calnexin is there and this EGF is introduced, they can see that TrkB is spread equally across this endoplasmic reticulum. However, when EGF is not introduced, most of this TrkB TrkB are within this. Most of this TrkBs can be found around this endoplasmic reticulum, whereas in the case of where this EGF is introduced, they are spread across and try to move to the cell surface. And in the case of this calnexin knockout cells, where no calnexin knockout or calnexin depleted cells, where no construct has been used, they can see the cluster formations in the case. And when this EGF is introduced, they do not see any improvement in the cluster formations. However, for the wild type cells, what they can see that there are less clusters formations that can be observed, and they can see that they can see the less cluster formations of this TrkB in the case of this where the calnexin wild type is being introduced. And however, similarly, for this construct, the calnexin is being mutated with CK2 phosphorylation site. They can see that TrkBs are spread across the endoplasmic reticulum. However, for these mutations at this serine at 563 sites, they can see the cluster formations are similar to this case of where calnexin is being depleted, where EGF is introduced or not introduced. So from this, they again confirm that it's the phosphorylation site at 563 or MAPK phosphorylation size plays a role towards this transport of TrkB to the cell surface. And these things are been quantified in this figure, where they can see that when in the case of this calnexin depleted cells, calnexin positive cells are the control. In the case of this calnexin depleted cells, they can see that it's the when this construct that use the serine at 563 construct that has mutations, construct of calnexin that has a mutations at 563 sites has more cluster formations, and it is statistically significant compared to the case where no compared to the case where this wild type calnexin is introduced, construct is introduced, or this calnexin with this CK2 phosphorylation sites is introduced. So this further confirms that the phosphorylation at serine 563 positions is important for the transport of phosphorylations at 563 serine positions of calnexin is important for the transport of this TrkB from endoplasmic reticulum to the cell surface. So that's all for this video, and if you need to understand more about their experimental results, how they have conducted their experiment in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.